Coronaviruses are a very diverse family of viruses. They have a large host range, which includes humans. However, the greatest amount of coronavirus diversity is seen in bats. Coronaviruses have spiky projections on their surface that look like crowns. Corona means crown in Latin, and that's how this family of viruses got their name. Most of the time, human coronaviruses cause mild respiratory illnesses, like the common cold. In fact, 4 types of human coronaviruses cause upwards to 30% of all upper respiratory tract infections that occur in adults. A new type of coronavirus can emerge when an animal coronavirus develops the ability to transmit a disease to humans. When viruses are transmitted from an animal to a human, it's called zoonotic transmission. Coronaviruses that make the jump to human hosts can sometimes cause serious illness. This can be due to a variety of factors, particularly humans' lack of immunity to the new pathogen. These viruses were first recognized as a distinct group because of their characteristic morphology. The envelope is studded with long spikes, giving the appearance of a crown or a corona. They are the largest known RNA viruses. Almost 2/3 of the genome encodes non-structural proteins that are required for transcription and genome replication. The nucleocapsids are long, flexible helices. The E2 protein or S protein is the anti-receptor, and we see mutations associated with altered antigenicity and pathology for this particular protein. The S protein can also mediate cell-to-cell fusion and fusion of the viral envelope with the host cell membrane. E1, or the M protein, the matrix protein, is an integral membrane protein. Only a small portion of the protein is exposed at the surface. It interacts with the nucleocapsid to form the envelope through budding. The single-stranded RNA genomes of severe acute respiratory coronavirus and Middle East respiratory syndrome coronavirus encode 2 large polyproteins, PPA1 and PP1AB, which are proteolytically cleaved into 16 non-structural proteins, including a protease and the RNA-dependent RNA polymerase, also a helicase and an exonuclease. An additional 9 to 12 open reading frames are encoded through the transcription of a nested set of subgenomic RNAs. We'll talk more about those in a minute. Coronavirus spike proteins are key determinants for virus attachment and entry into target cells. CoV-S, or the spike protein, is the receptor binding protein. The receptor for both SARS-CoV and SARS-CoV-2 is angiotensin converting enzyme 2, or ACE2, a cell surface zinc binding carboxypeptidase that is important for the regulation of cardiac function and blood pressure. ACE2 is expressed in epithelial cells of the lung and small intestine, as well as many other organs. CoVs are envelopes, so penetration occurs as a consequence of a membrane fusion event. After the envelope spike protein binds to its cellular receptor, membrane fusion occurs either directly with the host cell membrane or with the endosome membrane. To be activated for fusion, the spike protein must be cleaved at 2 sites, an event that's triggered by factors such as acid pH. The viral RNA genome is released into the cytoplasm, and the RNA is uncoded to allow translation of the 2 polyproteins, transcription of the subgenomic RNAs, and replication of the viral genome. Translation of the positive strand genomic RNA gives rise to a large polyprotein that undergoes proteolytic processing to generate an RNA-dependent RNA polymerase. Through the action of the RNA polymerase, a full-length antisense negative strand template is generated. Subgenomic messenger RNAs are synthesized from subgenomic strand templates. Translation of the subgenomic messenger RNAs gives rise to structural viral proteins. So let's look at this process a little more closely. Once viral RNA is released into the cytoplasm, it is used to synthesize a viral RNA-dependent RNA polymerase, which initiates transcription of viral messenger RNAs. The expression of all genes beyond the replicase gene occurs from a series of subgenomic messenger RNAs. What happens is that ORF1A and ORF1AB, which are at the 5' end of the full-length viral genome, and you see those indicated in blue in the figure at the top of the slide. These ORFs are translated to produce two polyproteins that are then cleaved into a number of proteins to form the RNA replicase transcriptase complex. This drives the production of negative sense RNAs through both replication and transcription. During replication, full-length minus strand copies of the genome are produced and used as templates for full-length positive RNA genomes. During transcription, a subset of 7 to 9 subgenomic RNAs, including those encoding all structural proteins, are produced through discontinuous transcription. In this process, subgenomic minus sense RNAs are synthesized by combining various lengths of the 3' end of the genome with the 5' leader sequence necessary for translation. These subgenomic negative sense RNAs are then transcribed into subgenomic positive sense messenger RNAs. Although the different subgenomic messenger RNAs may contain several ORFs, only the first ORF that is closest to the 5' end is translated. The resulting structural proteins are assembled into the nucleocapsid and viral envelope at the ER and Golgi. This is followed by release of the virion from the infected cell. So basically what we have are several subgenomic RNAs are produced. They're nested, so they have a common 3' end and poly A tail. Each messenger RNA contains all the nucleotide sequences of the next smaller messenger RNA plus one additional gene from the 5' end. Only the 5' proximal gene of each is translated. Also, each messenger RNA has an identical leader sequence at the 5' end. This sequence is not translated. Experimental evidence suggests that each subgenomic messenger RNA is transcribed from a corresponding subgenomic negative sense RNA. The negative strand subgenomic RNA templates are generated by discontinuous transcription of the full-length positive sense strand genome RNA. There are transcription regulatory sequences, or TRS, that the polymerase interacts with, and a full-length negative strand is also made. The most abundant subgenomic messenger RNAs are the smallest ones, and the least abundant are the largest ones. Note that the structural proteins needed in the highest number for virion production are nearest the 3' end of the genome. So if you look at the figure at the top, the full-length genome, you can see that the N protein, the nucleocapsid protein, is the one that's at the 3' end of the genome. So that is going to be the one that's made, the one that's needed in highest amount, highest concentrations, and we find that those subgenomic messengers are produced in highest number. After adequate viral genomic RNA and structural proteins have accumulated, the N protein assembles with the genomic RNA in the cytoplasm to form the helical nucleocapsid. The nucleocapsid then acquires its envelope by budding through intracellular membranes between the endoplasmic reticulum and Golgi apparatus. So the S, E, and M proteins are transported to the ER Golgi intermediate compartment, or ERGIC, where the nucleocapsid interacts with the M protein to generate the basic virus structure and complexes with the S and E proteins to induce viral budding and release from the Golgi apparatus. The viral replication cycle is completed when the assembled virion is released through exocytosis to the extracellular compartment. This figure shows you the complete replication cycle of the coronaviruses. The life cycle begins when the S protein binds to the appropriate cellular receptor. After receptor binding, the conformational change in the S protein facilitates viral envelope fusion with the cell membrane through the endosomal pathway. Then the virus releases RNA into the host cell. Genome RNA is translated and processed to form the viral RNA-dependent RNA polymerase. The polymerase produces a series of subgenomic messenger RNAs by discontinuous transcription and finally is translated into relevant viral proteins. Viral proteins and genome RNA are subsequently assembled into virions in the ER and Golgi and then transported via vesicles and released out of the cell. There are many different coronaviruses, some affecting just animals, but for our purposes we're going to concentrate on the coronaviruses in humans. The first human coronaviruses were described in the 1960s. There are now 4 endemic human coronaviruses known that circulate worldwide in the human population. In most cases, these endemic human coronaviruses cause relatively mild diseases of the upper and lower respiratory tract and are estimated to account for about a third of all so-called common colds in humans. Asymptomatic infections have also been described. In some cases, especially in immunosuppressed individuals, children, or persons with existing pulmonary diseases, progression to acute respiratory failure can also occur. However, this situation completely changed with the appearance of SARS-CoV. This virus caused serious human respiratory diseases in China in 2002 through 2003. Approximately 8,000 people were affected by the disease at that time, with a case fatality rate of around 9.5%. Human coronaviruses continue to pose a threat to human health. So since that first SARS in 2002, we've seen the emergence of 2 other important coronaviruses affecting humans. So we have MERS-CoV, which is Middle Eastern Respiratory Syndrome, and then in December 2019, SARS-CoV-2 became the third introduction of a highly pathogenic coronavirus into the human population in the 21st century. Bats harbor a wide range of coronaviruses, including severe acute respiratory syndrome coronavirus-like and Middle East respiratory syndrome coronavirus-like viruses. SARS-CoV crossed the species barrier into masked palm civecs and other animals in live animal markets in China. Genetic analysis suggests that this occurred in late 2002. Several people, close proximity to palm civex became infected with this virus. A MERS-CoV ancestral virus crossed the species barrier into dromedary camels. Serologic evidence suggests that this happened more than 30 years ago. Abundant circulation of MERS-CoV in dromedary camels results in frequent zoonotic transmission of this virus. SARS-CoV and MERS-CoV spread between humans mainly through nosocomial transmission, which results in the infection of healthcare workers and patients at a higher frequency than infection of their relatives. So let's look at some of these infections. So let's look at the first SARS. In 2002, a new human respiratory disease emerged in southern China. The following year, one of the doctors who had been treating patients traveled to Hong Kong, where he became sick and died. Subsequently, people in the hotel where he had stayed traveled to Singapore, Vietnam, Canada, and the U.S. Now the epidemic was underway. The causative agent was found to be a new coronavirus. Viruses with similar genome sequences were isolated from animals sold in markets in the region of China where the first SARS cases appeared. Many workers had antibodies but no history of disease. It is likely that coronaviruses have repeatedly crossed from animals to humans, but in the majority of cases, the virus lacked the ability for efficient human-to-human transmission. This original SARS outbreak was brought under control by quarantine measures. By executive order in 2003, SARS was added to the list of quarantinable communicable diseases in the United States. Currently, there is no known SARS transmission anywhere in the world. The most recent human cases of SARS-CoV infection were reported in China in April 2004 in an outbreak resulting from laboratory-acquired infections. CDC and its partners, including the World Health Organization, continue to monitor the SARS situation globally. Any new updates on disease transmission and SARS preparedness activities will always be well publicized. An interesting occurrence with this outbreak was the emergence of a superspreader host. A superspreader host is an organism infected with the disease that infects disproportionately more secondary contacts than other hosts who are also infected with the same disease. So in this case, a sick human can be a superspreader when they're more likely to infect others than most people with the disease. We're always very concerned about superspreaders in epidemiology. And in this case, you can see the layout of the 9th floor of the hotel where the doctor from China who was infected with SARS came and was staying. And we saw a number of people who became infected in this hotel. And you see in green, that was the primary case patient's room, but all the rooms indicated in blue were secondary cases who contracted it either from direct interaction with the primary case patient or from contaminated areas. So for instance, you can see the red oval near the elevators where they found contamination. It was a positive environment where they found samples, so people could have been infected there. And it was a very interesting situation, but again, it was brought under control and eventually eliminated through quarantine. We'll talk more about the difference between this first SARS and SARS-CoV-2, which we're now experiencing as a pandemic, in a few minutes. Severe acute respiratory syndrome is a viral respiratory disease of zoonotic origin caused by the SARS-CoV virus. And again, we had an outbreak. There was nearly a 10% fatality rate, but it was only by careful control that this virus was essentially eliminated. And it was kind of an interesting situation for my family, because my brother, who is also a PhD entomologist, he was taking part in the semester at sea program, and he and his wife were on a ocean liner that literally went around the world. It had students from universities all over the U.S. and faculty from many different universities, and they traveled around the world, stopping in various ports, and the faculty were providing various kinds of lessons and things on the geography, the ecology and biology of the different ports where they were going to stop. Well, this outbreak occurred right in the middle of their trip. And if you look in this slide here, you can see Taiwan, Vietnam, Thailand were all hot spots for the virus. Well, those just happened to be areas where the ship was intending to stop. So they had to completely rearrange the whole itinerary for this trip, because even though the risk of an individual from the ship getting off and contracting the virus in one of these ports was probably low, if somebody had contracted the virus and then came back onto the ship, you could have had another super spreader event like we saw in that hotel in Hong Kong. Middle East Respiratory Syndrome, or MERS, is a viral respiratory disease caused by another novel coronavirus. This one was first identified in Saudi Arabia in 2012. MERS-CoV is a zoonotic virus, which means that it's transmitted between animals and people. Studies have shown that humans are infected through direct or indirect contact with infected dromedary camels. MERS-CoV has been identified in dromedaries in several countries, including Egypt, Oman, Qatar, and Saudi Arabia. And MERS-CoV specific antibodies have been identified in dromedaries throughout the Middle East, Africa, and South Asia. The origins of the virus are not fully understood, but according to the analysis of different viral genomes, it's believed that it may have originated in bats and was transmitted to camels sometime in the distant past, probably at least 30 years ago. Approximately 80% of human cases have been reported by Saudi Arabia. What we know is that people get infected there through direct contact with infected dromedary camels or infected people. Cases identified outside the Middle East are usually traveling people who were infected in the Middle East and then traveled to areas outside of the endemic region. On rare occasions, outbreaks have occurred in areas outside the Middle East. Here you can see the signs and symptoms of MERS-CoV, very similar to what we saw with the first SARS and what we're seeing with the current, SARS-CoV-2 or COVID-19. So fever, cough, shortness of breath, pneumonia can occur. There can be gastrointestinal symptoms. Now, alarmingly, with this virus, it has a 35% mortality rate, very high mortality rate. This shows you the laboratory confirmed cases of MERS. between 2012, remember that's when the disease was discovered, and 2019. The median age of cases was 55 years old, ranged from 67 to 87 years of age. The majority of cases were men, 59%. 26 cases, or 14%, were in healthcare professionals. At the end of September 2018, a total of 2,260 laboratory-confirmed cases of Middle East respiratory syndrome, including 803 associated deaths, were reported globally. The majority of these cases were reported from Saudi Arabia. Owing to improved infection prevention and control practices in hospitals, the number of hospital-acquired cases of MERS has dropped significantly since 2015. Now, if we look at latest WHO statistics, this is through 2019. We've had almost 2,500 confirmed cases, with more than 850 global deaths, and 27 countries that reported cases. Now, we're seeing a reduction in the global threat of MERS because of addressing various knowledge gaps that existed with regard to transmission, also enhancing surveillance and strengthening the ability to detect cases early and contain outbreaks through improved prevention and control measures, particularly in hospitals. Critical for preventing international spread and sustained transmission has been improved prevention and control measures in hospitals, restriction of camel movement in affected areas, stronger and more comprehensive investigations of cases and clusters, and improved communication. Although global efforts seem to have prevented hundreds of infections and deaths, vigilance must be maintained by all countries. More needs to be done to limit spillover infections from dromedaries, which require stronger surveillance of dromedary populations and persons who are in direct contact with infected herds. We also need to develop a vaccine for the camels. An international community and infected countries have a collective and shared responsibility to address these needs. The largest outbreak outside of the Middle East of MERS occurred in the Republic of Korea. It resulted in 186 cases and 39 deaths. The recent exported case to the Republic of Korea in September 2018 did not result in further human-to-human transmission in the Republic of Korea. This was due to early suspicion of MERS-CoV infection, immediate isolation, clinical management of the patient upon his return from Kuwait, and thorough contact tracing. So that just shows you, if you look under 2015, you see that big spike in red, that in the Republic of Korea. That's when they weren't expecting MERS. The international community wasn't well versed in what to do, and in particular, because they didn't understand what the risks were, healthcare workers were at high risk of contracting the disease. Now, if you look under 2018, you see that one tiny little red dot. Knowledge is power in this circumstance. Listen to the science. They were able to suspect MERS, test for MERS, isolate the patient, and through these kinds of management techniques, even in the absence of a vaccine and a treatment, they were able to contain the virus and prevent an outbreak that second time. Here you can see what's going on specifically in the U.S. So we've had more than 8 million total cases and more than 220,000 deaths in the U.S. since the pandemic started occurring here. People with COVID-19 display a wide range of symptoms ranging from mild to severe illness. Symptoms may appear anywhere from 2 to 14 days after exposure to the virus. Now, I've listed here the symptoms that you should look for, and it doesn't list all of the possible symptoms. And, particularly as we learn more and more about the virus, additional symptoms may be recognized. And then when to seek emergency medical attention. These are not all-inclusive either, but it does list the most substantial ones. So if you're experiencing trouble breathing, persistent pain or pressure in your chest, confusion, well, in my case, more confusion than normal, inability to wake or stay awake, bluish tinge to the lips or face, those are signs of an individual who needs to get medical attention now. You should call 911 or call ahead to your local emergency facility. And you need to notify the operator that you're seeking care for someone who most likely has COVID. So this virus, officially named SARS-CoV-2, enters the body generally through the mouth or nose. From there, the virus makes its way down into the air sacs inside the lungs that are known as alveoli. Once in the alveoli, the virus uses its distinctive spike proteins to hijack cells. The primary genetic programming of any virus is to make copies of itself, and COVID-19 is very good at this. Once the virus's RNA enters a cell, new copies are made, and the cell is killed in the process, releasing new viruses to infect neighboring cells in the alveolus. This process can occur initially without a person being aware of the infection, which is one of the reasons why COVID-19 has been able to spread so effectively. There's an immune response. The process of hijacking cells to reproduce causes inflammation in the lungs, which triggers an immune response. As this process unfolds, fluid begins to accumulate in the alveoli, causing a dry cough and making breathing difficult. For more than 80% of people infected by COVID-19, these symptoms will run their course much as they would with the case of the flu. However, in 15 to 20% of cases, the immune system's response to inflammation in the lungs can cause what's known as a cytokine storm. This runaway response can cause more damage to the body's own cells than to the virus it's trying to defeat, and is thought to be the main reason why the conditions of young, otherwise healthy individuals can rapidly deteriorate. If enough alveoli collapse, a patient has to be placed on a ventilator for breathing assistance. Both acute respiratory distress syndrome, or ARDS, and high altitude pulmonary edema, or HAPE, are being investigated as causes. At this stage, the surfactant that helps keep alveoli from collapsing, surfactant is just something that essentially is almost like a lubricant inside the alveoli, and it keeps them from collapsing, because remember, this is where the air goes in and gas exchange occurs. So it's important that they remain open. You can see in the illustration, they almost look like a cluster of grapes. So if the surfactant becomes diluted by the fluid that's accumulating in the alveoli, and this fluid also often will contain cellular debris and all that, this all impairs gas exchange, which is the process that supplies oxygen to our bloodstream. In the most severe cases, systemic inflammatory response syndrome, or SIRS, occurs as the protein-rich fluid from the lungs enters the bloodstream, resulting in septic shock and multi-organ failure. This is often the cause of death for people who have succumbed to a COVID-19 infection. SARS-CoV-2 enters host cells through the interaction of its spike protein with the entry receptor ACE2 in the presence of TMPRSS2 as a co-receptor. Proposed mechanisms for COVID-19 pathology include, first, just direct virus-mediated cell damage. Secondly, dysregulation of the renin-angiotensin-aldosterone system, or RAS. The RAS is a hormone system within the body that is essential for the regulation of blood pressure and fluid balance. It is thought that viral entry disrupts this system by binding to the ACE2 receptor. Also, endothelial cell damage and thromboinflammation occurs. And lastly, dysregulation of the immune response and hyperinflammation caused by inhibition of interferon signaling by the virus, T-cell lymphodepletion, and production of pro-inflammatory cytokines, particularly IL-6 and TNF-alpha. Although COVID-19 is most well known for causing substantial respiratory pathology, it can also result in several extrapulmonary manifestations. These conditions include thrombotic complications, myocardial dysfunction and arrhythmia, acute coronary syndromes, acute kidney injury, gastrointestinal symptoms, hepatocellular injury, hypoglycemia and ketosis, neurological illnesses, ocular symptoms, and dermatologic complications. Given that ACE2, the entry receptor for the causative virus SARS-CoV-2, is expressed in multiple extrapulmonary tissues, direct viral tissue damage is a plausible mechanism of injury. In addition, endothelial damage and thromboinflammation, dysregulation of immune responses, and maladaptation of ACE2-related pathways might all contribute to these extrapulmonary manifestations of COVID-19. So clearly this virus is much more than just a simple flu or a simple respiratory infection. And here we've got a comparison between the first SARS virus, MERS-CoV, and COVID-19. And, there have been no global SARS outbreaks of the first SARS since 2004, and those last ones were acquired in a lab. There have been no more cases since then. That virus has been successfully contained using public health measures such as early case detection and isolation, contact tracing and isolation, social distancing. And with MERS-CoV, some of the same kinds of techniques have been put into place, in particular protections of healthcare workers to prevent nosocomial infections. And then, of course, we have the current SARS virus, COVID-19. There are reasons for why COVID-19 has been more difficult to deal with. Why haven't we been able to get it under control, for instance, as quickly as we did the first SARS virus? And there's some factors, some characteristics of COVID-19 that contribute to this. So first of all, people with COVID-19, probably about 80%, have mild illness. Some of them don't even know that they're sick. So this makes it harder to determine who's infected and who's not. Also, people with COVID-19 appear to shed the virus earlier in the course of their infection than people with SARS. This makes it more difficult to detect who has the virus and isolate them before they spread it to others. COVID-19 is now spreading easily within communities. This was not the case with the first SARS, which was more commonly spread in healthcare settings. So if you look at these side-by-side comparisons, and you go down to the bottom, it's interesting that the number of people infected, COVID-19, far outstrips MERS and SARS. You look at the number of deaths, again, COVID-19 is far ahead of the others. And yet, when you look at the case fatality rate, the estimated case fatality rate is under 5% for COVID-19. It was almost 35% for MERS and almost 10% for the first SARS. So despite the fact that this virus has a lower case fatality rate, we're seeing far more deaths associated with it. And this has to do with the characteristics of the virus, the fact that it is just so much more easily spread and transmitted than these other viruses.